very busy weekend. I totally understand where you're coming from.
Right. Okay, can you see the screen?
Yep.
Okay, so we are going to explain the bottom part of this figure. I encourage you to contextualize the relationship between cell respiration and photosynthesis whenever you can, and to try to roll together some of the themes that are important in both processes. That's like last unit, we sort of set that up. So, you know, things like endergonic versus exergonic, entropy, loss of heat, loss of energy, the relationship between products becoming reactants and reactants becoming products. That's all really important for this. So we are trying to make a lot of ATP in this endosymbiotic organelle, the mitochondrion. And in doing so, we release low energy products that can be used in other systems like photosynthesis. Okay. Same idea, but with a lemur. There you go. That was just for you guys. And then keeping them in mind, the idea that these are all just a series of redox reactions. Essentially, we're going to break down oxidized carbohydrates like glucose in the presence of oxygen. And when we do so, reactions occur, creating carbon dioxide and water. But we're really after this product of ATP in the end. Same idea, except with multiple arrows. You know why I like that one a little better? Yeah. Yeah, it's showing you more detail, right? So here I like this one because it brings up redox reactions, but here it's showing you, you know, there's multiple steps. You know, and in those three sort of steps, there's multiple reactions, right? So it's just accurate to say, hey, by the way, when we summarize this reaction, we're skipping over a fair amount of things going on, right? And so some students, you know, they're like, summarize cell respiration. And you could do it that way, but we're going to go into a little more detail there. So why is it called cellular respiration? Well, what does it mean to respire? Alex, what do you think? Yeah, breathing, bringing certain things in, certain things out, right? So it's like cellular breathing in that the cell needs to bring in what key gas to facilitate cellular respiration? Oxygen. And in doing so, it takes out a certain gas that's a byproduct of that series of reactions. What gas is that? CO2. So literally when you're breathing in and out, you're inhaling oxygen that goes to your mitochondria and you're exhaling CO2. That's a byproduct of the Krebs cycle. Does it take energy to do that? No, right? It's simple diffusion of small non-charged gases, right? It's pretty convenient. So it's just using concentration gradients to come in and leave the bloodstream. We had mentioned that earlier in the year. So this crazy molecule is NAD+. It's a lot like NADP+, right? And it's essentially what's the function of it. So obviously it's like a super complicated molecule. What does it look like? It looks like a nucleotide, right? So part of the name of it is dinucleotide, but you don't have to know the structure of this molecule down to like the, you know, the sugar, the nitrogenous bases, and the phosphates. Here's what I want you to take home. NAD+ becomes NADH when it takes on 2 electrons and 2 hydrogen ions. So I don't really even emphasize the hydrogen ions. I want to emphasize that when it's reduced, it becomes NADH. So we're really emphasizing that it takes on electron energy and transports it throughout the cell for various functions, which we'll get to in a moment. But Yeah, understanding the structure of it and that it takes on a hydrogen and some electrons is important, but you don't have to recognize it, you know, at this sort of level. Let's just call it NAD plus, okay? And it's when it's reduced, it's NADH. So this is a really important slide for this unit. So again, this anyone know how I can get rid of this thing from popping up whenever this comes over here?
I believe there should be a way you can drag that. At least I found that with Google Slides. You can sort of drag it off to the side.
This thing? This thing? Let's see.
Like it just goes away eventually.
It does go away, but I want to use this mouse arrow for a so every time I do that, though, it comes back.
I think I understand Dr. Kitzer, though. I appreciate it.
Okay. I mean, I have this for you guys, but that doesn't do him any good. What is this?
Dr. Kitzer, I think I understand.
Okay.
Yeah, I'm cool.
You see, I'm doing this literally just for you. So if you're good, then we're good. But that's the kind of service I offer.
5-star service indeed.
Yes. It's a 1-to-1 teacher to student ratio in this virtual classroom. Okay. So this organelle again is mitochondrion. Now they're drawing this coming from 2 cells. Why is that? Because they both have it. I keep going back to that because don't fall for that, right? Like, yes, photosynthesis has chloroplast in autotrophic cells, which is not present over here, but they both need to break down those molecules, right? So it's coming from both of these eukaryotic cells. Now, let's just examine this figure and bring some takeaways from it, even though we haven't gone into the lesson. So glycolysis, preparatory reaction brings pyruvate into the matrix of the mitochondrion where the citric acid cycle occurs. Then we see these folding of the inner membrane where the electron transport chain and chemiosmosis occurs. I know I'm throwing a lot at you, but let's break it down. What does that mean about glycolysis? Tell me one thing that you can say about glycolysis from this picture. Yeah. Good, so you can say like the initial reactant is glucose, the final product is pyruvate for this particular series of reactions. What else though? Yeah.
Won't you also make ATP?
You make a little bit of ATP. That figure down here is showing that you net 2 ATP in that reaction. Anything else? Might not be so obvious. We're also taking some electrons and we're sending them to the ETC in the inner membrane. But there's one thing that's not obvious in this picture. Where does glycolysis occur? Not in the mitochondrion. So outside the mitochondrion, which technically, what is all this stuff? Cytoplasm. So glycolysis does not take place in the mitochondrion. It takes place in the cytoplasm. So what does that mean from an evolutionary standpoint, since we're talking about prokaryotic and eukaryotic cells, and prokaryotes not having internal membranes or internal organelles like the mitochondrion?
I think that, the only, the only species are the only people that are able to absorb and run that citric acid cycle and produce a lot of ATP to run a lot of functions is most likely going to be it's going to be eukaryotes because they have that mitochondrial membrane. Meanwhile, glycolysis is most likely happens in prokaryotes, and yeah, a little bit of ATP is produced, but of course not enough to drive a human or something like that. And there's also connections with fermentation too.
Excellent. Yeah. So Basically what we're saying here is glycolysis is the most primitive form of energy production. And we inherited that from our prokaryotic ancestors. And he's also linking it to processes like fermentation, which occur in an anaerobic environment, which also predates aerobic respiration. Okay, so there's a lot of evolution connections when it comes to glycolysis and energy production. We will be talking about lactic acid and alcoholic fermentation at the end here. So keep that in mind when we talk about glycolysis. Just a little bit of ATP, not enough to run a multicellular complex organism, but certainly enough for a bacterium, okay? Now, pyruvate needs to be converted into something before it can participate in the cyclical part of the citric acid cycle. That's what that's showing. Keep in mind, two membranes, again, supporting the endosymbiont theory. We're getting some electrons, again, going to the ETC. Anytime you see an electron, where is it going to go? To the ETC in this inner membrane, okay? So citric acid takes place in not in the space in between the membranes, but in the center of the organelle, what we call the what? The matrix. So has anybody seen the movie The Matrix? If you haven't, it's a must. A lot of times when I ask people your age if they've seen The Matrix, they're like, no, what is that? And it makes me a little sad. If you haven't seen The Matrix starring none other than Keanu Reeves, you should. I don't know, about 2 and 3 is kind of, Okay, so then it's say another thing about the citric acid cycle. What's another thing that we can see from the citric acid cycle from this picture? Or so it's occurring in the matrix. Does it make any ATP?
No.
It actually does make a few molecules of ATP. Not enough to write home about, right? Obviously, the last part of our process is going to make the most ATP, but a little bit. Does it contribute any electron energy to the final part of cell respiration? It does. We're showing all this like feeding in, right? So you get a little bit of NADH from the PREPS reaction. You get a little bit of NADH and FADH2 from the citric acid cycle.
Dr. Kinzer, could I ask you a question? I just had something that I was curious about evolution. So, you know, we know that all these little reactions, like the preparatory reactions and the citric acid, reactions, all contribute some ATP. Do you think, maybe those are like steps in evolution? Maybe the first step, coming from a prokaryote, maybe it ran glycolysis and the preparatory reactions. And the next step was maybe running all 3 reactions. And then maybe the most advanced, or what we have right now, is running all those 4 reactions and producing the most ATP.
I think that's fair to say. I think it's a safe assumption that on the way to increasingly complex, you have intermediate steps of complexity. Yeah, I think that's rational.
Okay, I was just curious.
Yeah, that's good thinking. What is not depicted in this figure as far as products go? It's showing all the useful, highly potentially energetic molecules and electrons that are being produced, but what is it not showing? Remember that respiration thing, Pratik? Yeah, it's not showing CO2 produced, which is a byproduct of citric acid cycle. So keep that in mind. I mean, we're gonna show it in the next figures, but okay. And then the last part is where you perform. But here's the good news. The last part you already know because we've covered it in photosynthesis. It's just now occurring in a different membrane, right? We're going to talk about electron transport chain and chemiosmosis and how you really crank out a lot of ATP at this last part because we've taken energy from previous steps and really made the process push towards occurring, right, and producing ATP. Okay. This picture I like a lot. Well, it depends. Like you're getting, the picture of the mitochondria is a little more 3-dimensional in the other one, but we're basically summarizing a little bit of ATP, a little bit of ATP, a lot of ATP. some electrons, they all go here, outside the mitochondrion, in the matrix, in the inner membrane. So here's the outer membrane. Here's the inner or interspace, the space in between the membranes. This is the inner membrane, and this is the matrix. Okay, so I want you to know where all these processes occur in those spaces. But first, let's watch a quick video.
As this mountain biker heads up,
Sorry.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvate acid, which still has a lot of energy available. Let's follow this pyruvate acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, one carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain. and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondria, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Wow. 10 million per second per cell? That's almost impossible to actually fathom, right? I mean, the volume of molecules that can be assembled inside of our cells. Okay, so there's a lot of themes to grab onto as we do that. If I had to summarize the entire process though, think about, you know, if you were to take a car and strip it for parts, okay? So you have a nice car and you're just gonna strip it full of all of its parts, right? So, you know, you might pass it along to, you know, the next person for the removal of the parts. Maybe the first person takes off the tires, maybe the second person, you know, takes off the windshield wipers or whatever. Each time you do that though, you're extracting some energy, but there's still some energy to be stripped away from the molecule, right? Until the very end. So I just see this as, you know, you're taking something that has a lot of stored potential energy in the molecule, right? A glucose molecule, which we've used energy to make it that way, right? We've used an endergonic process to make this happen. Now we want to release the energy, but we're going to do it very slowly, right? It's not like a one sort of one reaction and you've taken all the energy. Why is that the case? Why do you think that we have, you know, we're going to talk about glycolysis here next. Why do we, you know, when going from glucose down to pyruvate, all these different reactions, and then you have the Krebs cycle and all the different reactions. Why do we separate it into so many different reactions? Think about this, contextualizing it with the previous energy unit and how entropy works. Sure, I mean, we're doing that, you know, throughout the whole series of reactions, but every time you go from this reactant to this product, and if that product becomes a reactant, you go from this reactant to this product, what are we losing every step of the way? Not necessarily ATP, but some energy in the form of heat, right? So not all these reactions are 100% efficient. So here's a related question. If you come across a house in the woods, abandoned, nobody lives there, you know nobody lives there, it's freezing, you're not going to make it unless you find some sort of shelter, okay? Or how are you going to stay warm? Now, whenever I ask this, people are like, just go inside, dummy, right? Like, it's a house. You can't go inside. You can't go inside. You've decided that you have to start a fire to warm yourself up to make it through the night. How are you going to do that to make it through the night? Do you just set the house ablaze? You do.
You take off pieces of the house.
Yeah, so why do you take pieces of the house off?
So it lasts longer.
How would it last longer?
Well, if you just like burn all the wood at once.
It sounds like a dumb question, but yes, you're right? So if you start one small continuous fire by slowly stripping the house of boards, you still won't use all of the heat from the fire. Some of it's going to get lost as light. Some of it will go and it won't warm you at all. But certainly if you set the entire thing ablaze, you'll lose even more and you won't have that for a longer period of time, right? Does everybody get what I'm saying? Like by stripping it down, and converting it into intermediate products, you're able to collect more of the energy from the molecule over a period of time than you would if you try to do one large reaction to grab the energy, right? And so, and along with that, we have cycles that use some of the leftover products and feed it into the next part. So, you know, by having glycolysis, the Krebs cycle, you get more out of the molecules, right? You're powering the ETC with more electrons over the time period. Okay, so let's talk about glycolysis. What does glycolysis mean? Break down that word in your mind. Let me change the pronunciation. Glycolysis. Splitting of sugar. So you have glucose here. What's happening to glucose in glycolysis? Well, how many carbons are in a glucose? 6. Okay. How many carbons do you think are in a pyruvate? So you have two 3-carbon molecules, right? So it's just been split. Well, there's an energy investment phase. The video talked about it actually costing you some ATP to get the process going. What's up with that? I thought we were trying to make ATP. And now all of a sudden you're like right off the bat in a -2 get. What do phosphates do to molecules? Thinking back to the last unit on energetics.
They charge them.
They charge them. What's another way of saying they're charged? They're energized. What's another way of saying like energized? They're phosphorylated. They're more reactive. They're less stable, right? So now once you have phosphorylated the molecule, it's unstable. It's more likely to participate in chemical reactions. It breaks. You know, you're on your way to, gotta stop touching this thing. You're on your way to making some energy. So that initial 2 ATP investment ultimately is going to yield 4 ATP, but it'll net 2 ATP in the end, right? So you basically pay back your energetic debt and you actually make 2 ATP. How does that work? The video actually showed this. So if you've now phosphorylated a molecule, how can you make ATP from that molecule?
You take it, phosphate.
Yeah, you take the phosphates back.
Yeah.
Yeah, so this is an interesting phenomenon that's different than the ATP production in the last step of the process. This is a process called substrate level phosphorylation. So it's going to take a phosphorylated substrate and it's going to put both this and an ADP into the enzyme. And that enzyme can facilitate the transfer of the phosphate from the substrate to the ADP, making ATP. So it's called substrate level phosphorylation. And that's actually how you make your ATP back in glycolysis. It's not through the ATP synthase, it's substrate level. And actually the same goes for glycolysis, Krebs cycle as well. What else do we make in glycolysis? And by the way, I've completely oversimplified this series of reactions on purpose. I can show you what they look like in a moment, but I'm just trying to get like at what the products are. What else do we make besides netting 2 ATP? Tell me about those electron carriers, the NAD+. They kept showing up to the molecule and taking something. What were they taking? Electrons, yeah. So, and I'm happy with that explanation of it. They actually take hydrogens, but so I want to emphasize the electrons though, because they end up dumping some of the protons down the road. But yeah, they pick up electrons and take the electrons where? ETC. So anytime I say like they're taking electrons, where are they taking them? You can say that. And that's sort of intuitive, right? They're taking them to the electron transport chain. And then what else do they make? What is left over after you've reclaimed the phosphates through substrate level phosphorylation. You're left with a 3-carbon molecule called pyruvate. Okay, so here's what you net. I'm gonna let you take your pick when it comes to potential energy. You can take the ATPs, you can take the NADHs, or you can take the pyruvates, okay? And that's your payout. Which ones are you taking? This is very similar to that question that I asked you, rank the potential energy in the molecule. What's your best bet? So we have ATP, pyruvate. Anybody gonna make a claim for the NADH? I thought NADH powers the whole thing. See some people not voting. Don't forget to vote. Raise your hand for ATP.
Are we talking about just like from glycolysis? Like the ATP from like
Yeah, I'm saying here's your final payout. You can get pyruvate, ATP, or NADH. What are you taking, Petit?
I think I'm taking pyruvate.
Okay. Now you guys are all taking pyruvate?
I said pyruvate. He said that, I say it too.
And don't vote that way. Just because your friend's voting that way, you can vote however you feel.
ATP is not worth it.
ATP is not worth it?
Only 2 ATP.
ATP 2020. There we go.
That's right. Powering the cell since,
Yeah, I'm not about ATP.
Okay.
ATP.
So like to answer this question, again, you have to understand this idea of like potential energy and like think of the long-term end game. So if you're cashing out on the ATP or the NADH, it's kind of short-sighted. Why is that? Where is this pyruvate going next? To the Krebs cycle, right? And so it can like process and break down that molecule, further extracting energy, right? So it's like if somebody says, I don't know, I could pay you what I owe you, $100. Or you can take this, I don't know, box of worthless baseball cards. And like you go through the box and it's like 95% worthless cards, but there's like 5 cards in there that are, I don't know, this is a weird analogy, but like worth thousands of dollars, right? I mean, this is in the long run, you might not be able to do anything with it right now, but as we process it through the Krebs cycle, it's going to be pretty valuable from an energetic standpoint. Here's another picture of substrate level phosphorylation. And I want to show this because it's very interesting. Does BPG and 3PG sound familiar? You remember it from the Calvin cycle? Okay. So what's going on there? When you take a phosphate from BPG, it becomes 3PG. Is that what that's saying? Do you remember the order of products in the Calvin cycle? 3PG led to BPG, right? So it's like we're undoing what we did in photosynthesis, right? And then eventually we made G3P. So look at the actual process of glycolysis. So we have the energy investment phase up here, which requires some ATP, ends up splitting the molecule into 2. So you have your phosphates on either side because of the ATP. And then we sort of process through, and it goes from G3P. Remember we put 2 G3Ps to make glucose? Now we're going in the reverse direction, right? We're taking that glucose that we made and we're breaking it back down into those products. Yeah.
Do we need to know the names?
No. So the take home in the previous slide is like you're investing some energy, you're extracting some energy, you're net positive as far as energy goes, but you're left with pyruvate, which is, useful for the next step. But I just think it's interesting, we're going back in the reverse. So you're just breaking down that molecule that you endogenically constructed, and you're breaking it down and releasing energy slowly, right? Okay, so yeah, you go from BPG, 3PG to PEP, down to pyruvate, and you're reclaiming the ATP through substrate level phosphorylation. Now this pyruvate goes on to, okay, so yeah, we'll talk about fermentation. Fermentation is just a modified glycolysis that makes different byproducts. So instead of ending at pyruvate, they end in alcohol or lactic acid production. But we'll talk about that at the end. Okay, and there's a preview of it. Okay, so now we're moving on from glycolysis to the second stage of cell respiration, which has two big parts. The first big part is this preparatory reaction. So pyruvate needs to come in from the cytoplasm into what space? Into the mitochondrion, but specifically matrix. So look, we've crossed the outer membrane, the inner space, the inner membrane, and now we're in the matrix. So keep in mind that the whole purpose of organelles and subcellular compartmentalization is to house enzymes for certain reactions in certain spaces. So we have a little more control over them. So pyruvate, again, a 3-carbon molecule, ultimately is converted into what? Follow that arrow. Yeah, it's acetyl-CoA. How many carbons does acetyl-CoA have? 2. So that means that we, what? Lost one. So we lost one in the form of carbon dioxide. So a molecule of carbon dioxide is literally removed from the end of pyruvate, right? So we're left over with this. You can see that over there. So that's step 1. You remove CO2. What else must happen? Remember, each step, each reaction, we're going to take electrons essentially. So at each step, NADH is made. Where does NADH go? To the ETC in the inner membrane. So I guess technically it goes here. And then we're left with acetic acid. So that's acetic acid and doesn't become acetyl-CoA until we do what? Plop that on there. So this little squiggly line, do you think this means that it stays on forever or that it falls off? Falls off? Yeah, so this coenzyme A is only on the molecule for a short time. I'll tell you what it does in a minute. So what does adding something like a coenzyme A or actually any other collection of atoms do to a molecule? What have we seen when we phosphorylate things? Destabilizes. Does that make it more or less reactive? More reactive. So if you're looking at this process, acetyl-CoA is energized just enough to join into the cyclical part of the citric acid cycle. So what's left over from previous reactions, oxaloacetate, over here, can join with this now energized molecule to make our first product of the citric acid cycle, which is what? Citrate, good. Which is why we call it the citric acid cycle. Okay. Does everybody get that? So the coenzyme A is just destabilizing the molecule enough to encourage it to join together with oxaloacetate to make this first product. Okay. So again, pyruvate, CO2 comes off, electrons are stripped, CoA goes on. We have acetyl-CoA, coenzyme A falls off once citric acid is citrate is made. Okay. So to summarize that little intermediate step, these two pyruvates ultimately become acetyl-CoA and 2 CO2s. You could also say that there's a NADH produced as well. Where does all this happen? Matrix. Okay. So going from citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate, you don't have to know those. okay? But you should be able to tell me something once I show you these carbon skeletons. You should be able to tell me some key events, okay? So for example, this is our first product right here, citrate. It becomes isocitrate. How many carbons are in citrate and an isocitrate? Can you guys see that from there? No, there's 2 from here. There's 4 here. So you should have how many? 2. So there's 1, 2, 3, 4, 5, 6. Here there's 1, 2, 3, 4, 5, 6. So is carbon dioxide produced in that reaction? No. So there's no carbon dioxide produced in that reaction. What about going from isocitrate, R6, to alpha ketoglutarate? Well, there's 1, 2, 3, 4, 5. Yeah. So carbon dioxide is produced in that reaction. Anytime you produce carbon dioxide, you're also taking some electrons. So you make a molecule of NADH. NADH goes where? ETC. To the ETC. Right, and so on and so forth as we go around. Now, you don't have to memorize this, so just keep that in mind, but you should be able to tell me what's happening in each step if I give you a picture of this, right? Carbon dioxide produced again, NADH produced again. Oh, we have another coenzyme coming in here. That must mean that going from alpha ketoglutarate, from succinyl-CoA to succinate requires a little bit of an additional energy, right? So there it is. We also see that because there's some ATP, right? So we're actually going to take some energy from that. Anytime you see GTP in GDP, we're not going to talk about it. It's like related to ATP and ADP, except instead of an adenosine, what do you think the G stands for? Guanine. Good. So in going from this reaction to this reaction, we actually take some energy. That's that 1 ATP that's produced per pyruvate in the citric acid cycle. Tell me something about succinate to fumarate. Anytime you see an energy box, there's what collected? Yeah, so FAD is an electron carrier like NAD+. So it's going to take some electrons and hydrogens, hydrogen ions, and it's going to make FADH2. Okay, where does FADH2 go? To the ETC, right? And so we go from fumarate to malate back to oxaloacetate. So you can literally count 1, 2, 3 NADHs, an FADH2, and an ATP per what molecule? Per acetyl-CoA. And you got the acetyl-CoA from? Pyruvate. So per pyruvate, you get that many. So if I could say per glucose, what's the number? Well, it's all this times 2, right? Because you get 2 spins of the cycle per glucose because you're making, you have 2 pyruvates that could go around. Okay. Here's the summary of it, right? They summarize this much better here. So we came in with a what? It's a 2 plus 4. So we have a 6 carbon molecule to start, 2 are lost. So without looking at oxaloacetate, how many carbons are left on it? 4, right? And then around and around and around it goes. Good. So I'm not showing you this, but you can get to acetyl-CoA from other non-carbohydrate means, okay? So by chopping up the fatty acid tails of lipids, or by converting amino acids into another form, you can get acetyl-CoA from those molecules. You don't have to eat carbohydrates to stay alive. Our friend Mr. Gallagher was evidence of that, right? So he wasn't following this glycolysis to citric acid cycle pathway. He was following an alternative means, right? So other metabolic pathways that break down macromolecules feed into this at the acetyl-CoA level. Okay, we'll mention it briefly at the end. I'm definitely showing you the traditional carbohydrate pathway, but it stands to reason from Mr. Gallagher being alive that you can get to this step through alternative means. And as long as you're doing this and creating electrons to power the ETC, you're going to be good. Okay. Any questions so far? A lot of stuff today. You guys all know that there are videos on my YouTube or anybody's YouTube that go over the steps of these reactions, right? Photosynthesis, light reactions, Calvin cycle, glycolysis, citric acid cycle. It's all out there. You can either watch mine or someone else's. I don't care. Yeah.
Yeah, I was gonna ask about that. Like, do the videos that you put up, are they for us or for like your freshman class?
So
do they go into enough detail for this class?
I would say that probably 70% are for the freshmen.
Okay.
And then 30% are for situations like this where students were like, can you run it through again?
Okay.
Right? So those videos would be good enough. And if, by the way, if there's a video, if there's a need for a video and the video is not there at the AP level, I'm happy to make it. You know, this weekend, somebody emailed me, I was like, I don't understand ET50. I'm like, I've got 20 minutes. I can make this video quickly. So, you know, don't be afraid to ask, but the videos that are useful for this class should be labeled AP.
Okay.
So if it says AP, it should be good. But you know, like even still, like some of the freshman video are helpful because I'm not requiring, you know, this level of knowledge, right? Which is sort of the college level. I'm just sort of emphasizing the themes that you need to take home, like, you know, redox reactions, you know, creating electron carriers. Okay. So, you know, I should be able to point to any of these and ask you for like themes that explain how they got there, right? Okay. Here it is shown in another way. Again, we're glossing over reactions by just showing some of the intermediates. So basically you put in, you know, a couple of low energy molecules. So low energy NAD+, low energy FAD, low energy ADP, and the Krebs cycle energizes those by breaking down higher potentially energetic molecules in each reaction, which creates high energy NADH, high energy FADH2, high energy ATP. Is carbon dioxide energetic at all? No. So it's not all highly energetic. Some of it's like toxic waste. CO2 you got to get out of your cells. Okay. You guys remember ETC from the other day? This idea of an uncontrolled reaction is what I was talking about before with the house. So you could just release all the energy at once, which is what an explosion is. Or you can kind of control it and slowly release it by burning board by board, which is what we do with the ETC and chemiosmosis. Okay, so we're gonna review this again, but in the context of cellular respiration. You know what, I'm gonna stop there. And I'll give you guys a quick quiz to see where you're at with your understanding from today. Do you have any questions before the quiz? Does anybody want to throw any ideas for your classmates of what they thought I emphasized? Yeah, take a quick glance down at your notes. Share out with your friends.
Each step contributes towards the ETC. So like electrons like get transported to the ETC, and a little bit of ATP is made through each step.
I would say that, yeah. So you're making, in the first two processes, you're making some ATP through substrate level phosphorylation. Nothing to write home about, but it's something. You're collecting electrons at each step, which ultimately power the ETC, which is going to lead to that massive production of ATP. What else?
Formation of pyruvate glycolysis. There's like Electron heavily.
Good. So glycolysis out in cytoplasm, it's going to result in the production of pyruvate, which still has a lot of potential energy in it, right? We just stripped a little bit of energy from pyruvate, going from from glucose to pyruvate. There's still plenty more to be extracted from it in the Krebs cycle. We make a little bit, but don't poo-poo pyruvate. Don't sleep on pyruvate. It's got a lot of energy. What else? Yeah.
Maybe like how in some steps you use pyruvate, but also you use pyruvate kind of as a reactant, but also get energy from it.
So pyruvate is the product of glycolysis, but it's the reactant of Krebs cycle. Yeah, isn't that a theme like in biochemistry, especially like metabolic reactions where you're going from one reaction to another? What is a product in this reaction becomes a reactant in another, for sure. And it's all going downhill, right? Like it's all like, well, let's say that there's 4 steps. The first reaction that results in a product, you know, it has that product still has a lot of potential energy in it, which will power the next series of reactions, right? And so it's, you know, you're feeding sort of downward energy-wise, but you're still extracting some energy from those reactions. Okay, I think we're good. I think 10 minutes is plenty of time. I think we had a nice little productive review. So, any questions about reading? You could see online that we have 2 classes for cellular respiration. So, how many sections are there in the book? 4?
For cellular respiration.
Yeah.
7.
7? Okay. Well, 7.
Yeah, I started reading the book and it was so long and there was so much detail, so I just watched your video. So I was wondering if that was like.
Was that useful for today?
Yeah, for sure. So, I was wondering if
I
needed to know more information.
Yeah.
The book is just so detailed.
The book is so detailed. They provide a lot of context and stories and experimental context.
Like for cellular respiration, your videos should be enough.
Yeah.
Plus whatever we're talking about in class.
Plus, contextualizing into previous discussions like, entropy, endergonic, exergonic, for the test, for this conversation, the video, correct me if I'm wrong, but it was just probably the same thing in like worse handwriting.
Yeah, probably. I mean, I didn't get the prep cycle.
Yeah, okay. And I need to remake those because I used a weird pen back in the day. So whatever, did you get to you got through section 2, section 3? Yeah. So it's kind of like a middle point. Do you feel like we covered that amount? Yeah. So sometimes I'm bad. Sometimes I'll put make sure you read chapter 7. And I forgot to say sections 1, 2, and 3. Sometimes people are like, well, what should I read? Half of it.
Yeah.
I don't know. Read it all. If you can, read it all. Get as far as you can, and I'll be a reasonable person. I'm not going to do. If I'm giving 2 classes to it, I'll have to stop somewhere in the middle. So I don't know. But keep in mind, and you guys are kind of lucky because I used to give these quizzes at the beginning of class. I stopped doing that. So I used to walk in and be like, "What did you read last night?" And give them a quiz. Now I'm like, okay, let's talk about it. Let's talk about what we read last night. Okay.
Dr. Kinzer, just wanted to make sure you'll be sending this quiz to me, correct?
Yeah, I will.
All right, sounds good. Dr. Kinzer, could I head out of the Zoom meeting, please?
Yeah, go ahead.
All right. See you soon.
I love how you asked for it. You will be sending
It really is like a 40-year-old.
And it's odd, isn't it? That's actually odd.